Hello everyone. In this video you are going to look at how to conduct a chromatography experiment with food dyes and two different solvents. And we will also look at some variables and differences in results when you compare chromatography. All right. What you see in this picture are the apparatus that you will use for a chromatography experiment. All right. So what you will use is boiling tubes. You will have two boiling tubes because you will do two sets. A pair of scissors, ruler, a marker, pencil, two chromatography papers, food dye, and deionized water and isopropyl alcohol. Okay, step one. This is what you would need to do. Use a pencil to draw a line at one end of the chromatography paper. Okay, you will need to draw the line about 1 cm from the bottom edge of the paper. Okay. Next, you need to cut the pencil line into a triangular tip. Okay, so this is how you do it. Okay, next, you need to use the splint provided or the stick provided to place a small spot of purple dye into the pencil line. And be careful not to take too large a drop because the separation will not be clear if you put too much food dye to start with. So dip it inside the food dye, shake off any excess drops that's coming. Make sure there is no large blobs on the stick. And then, yeah, place it on the pencil line. Okay, so this is how it will look like after you place the purple dye drop on the pencil line. So make one more of the same chromatography strip. So you should have two purple dye spotted chromatography papers. Now you need to use a split cork to secure the chromatography paper and fit it into the boiling tubes. So this is how a split cork looks like. Remove the rubber band, okay, and you will see that it will fall in two parts. So place the chromatography paper between the two halves of the split cork and then tie it back with the rubber band. All right. Okay, next you need to prepare two boiling tubes. And inside each boiling tube, fill one with distilled water and the other one with isopropyl alcohol. If you are only doing a distilled water experiment, then fill both boiling tubes with distilled water. So this is how you need to fill it with distilled water. You need to fill up about there. Okay, actually the idea is to make sure that the amount of solvent is below the pencil line in the chromatography paper. And the chromatography paper must be dipped into the solvent. In this experiment, we are doing isopropyl alcohol and distilled water. So distilled water in the lab is also called deionized water. So this is a bottle of deionized water you'll find in the lab. And fill the boiling tube to about that much of deionized water. If you need more, you can add more later. Right? Okay, so in this part, you will notice that the chromatography paper tips will not touch the solvent. So in this case, you need to top up the solvent so that the tips of the chromatography paper touches the solvent. Okay, so take your papers and put them in vertically and straight into the boiling tube and secure the split cork to cover the boiling tube. So in this case, you can see that the tip is barely touching the solvent. So I would have topped it up to the red line that you see. So that's how much solvent you need to put so that the tip of the chromatography paper is submerged into the solvent. But the pencil line is well above the solvent. Okay, this is going to be a video, fast forwarded time lapse video to show you how 20 minutes of chromatography, standing in chromatography looks like. All right, so here you go. So this is what will happen as you wait. You will see that the dye will travel up together with the water. In this experiment on this side, this is alcohol, and you can see alcohol travels much slower and the separation is not as clear yet. Right? So before the solvent reaches the end of the split cork, you need to stop the experiment, and that takes about 15 to 20 minutes. So over here you can see three colors: blue, yellow, and red. All right. Now for the alcohol one, the solvent only reached to about half of the paper. If you have enough time when you're doing with alcohol, you can allow another 15 to 20 minutes, and that will cause the chromatography the solvent to go up the chromatography paper up till the edge of the split cork. Okay, after you have allowed the solvent to run up the chromatography paper and you see a distinct separation, you can remove the paper from the solvent. Okay, but then you remember you need to remove the paper before the solvent front reaches the split cork. Now, when you take the paper out, these are some things that you will see. In this case, you can see that one paper was put in distilled water, the other one was in isopropyl alcohol. The blue ink is the most soluble in distilled water because it traveled the furthest. The same for the one in alcohol, the blue ink traveled the furthest, so it's the most soluble. In the one in distilled water, yellow ink was next and red ink was the least soluble because it was the lowest and traveled the least distance. However, in alcohol, the red ink traveled higher up and the yellow ink is the one that was the least soluble and traveled the least. All right. One more thing that you will note is on the pencil line, for the one on distilled water, there is nothing left on the pencil line. All the dye that we put on the pencil line has traveled up, which means every part of the food coloring is soluble in water. However, the same food coloring, which was soluble in water, does have some components that is still insoluble in alcohol, and that's why you see this gray spot over here, which means some parts of the dye has remained on the pencil line because it is insoluble. Okay, this is a comparison of green dye in isopropyl alcohol and in distilled water. So you can see green dye, which is made up of blue and yellow dyes, does not separate well in distilled water. Both the blue and yellow dyes traveled up together and they have similar solubilities because they travel the same distance. However, in isopropyl alcohol, you can see the blue and yellow have distinctly separated, which means isopropyl alcohol is a better solvent to separate the inks in the green dye. Right? This is the chromatography results of a black marker ink in water and in alcohol. So again, you can see the separation. Both alcohol and water were able to separate the different colored dyes in the black marker. Okay, this one shows what happens if you draw this pencil line with a marker instead. All right. So you can see the first one is the line is drawn with a pencil, so only the dye has traveled up. One more thing to note is this is what will happen if you put too much dye on the pencil line. So the spot will be too large and the dye doesn't really separate as it travels up the chromatography paper. On the other hand, when you draw the pencil line with a marker instead, the marker can also dissolve in the solvent, which will then travel up together with the ink, and you will have this smudgy uh, chromatogram that is not clear. All right. Okay, so that is the end of the chromatography experiment. Uh, do watch this video one more time, especially to know the steps of setting up your chromatography experiment.